It is my distinct pleasure to start next by introducing Dr. Richard Nowak, who is the chair of the MGFA Medical and Scientific Advisory Council, our chief medical officer, and a board member. Dr. Nowak?
Great. Thank you, Danielle. Let me just open up this slide. So, first of all, I'd like to thank everyone for their attendance, welcome you to the scientific session this morning, and introduce you to our keynote speaker, Dr. Ryan Hibbs. Dr. Ryan Hibbs is a professor and chair of the Department of Neurobiology at the University of California, San Diego. He and his laboratory focuses on the structural mechanisms of neurotransmitter receptor function and relationships in disease pathology. He's also a recipient of the High Impact MGFA Pilot Award and will be speaking to us about his work in the field of myasthenia gravis and the translation of structural biology and receptor structure and as that relates to disease mechanism today. So with that, I'd like to welcome Dr. Hibbs to the podium to deliver our keynote for this year's scientific session. So a warm welcome to Dr. Hibbs today.
All right, good morning, everyone. Thank you, Richard. Thank you, Danielle, for having me. Thank you to all of you for joining this early session in the morning for some basic science to wake you up. Right, so I come at MG from the perspective of trying to understand three-dimensional structures of proteins. That's kind of what I'm passionate about, and specifically neurotransmitter receptors. And we work on this superfamily of ligand-gated ion channels that includes nicotinic acetylcholine receptors and GABA A receptors, and we spent, I don't know, a decade trying to figure out how to make these proteins in quantities so that we could get high-resolution structures of them, first with X-ray crystallography and then cryo-electron microscopy more recently. And then only in the past few years have we turned our attention to disease, and we started partnering with clinicians to get, in this case, samples of autoimmune antibodies that we can then bind to the receptors and get structures and interrogate disease mechanisms coming from the atomic level. And so that's what I'll tell you about today. Okay. Ooh, here we go. Let's see. We have someone's cell phone up here that's beeping. Here we go. All good. Okay. We have funding from the MGFA. Thank you so much to the MGFA for all of this, bringing me to this meeting. And one of the most wonderful things about working with the MGFA for me so far is it's put us in touch with patient support groups, and getting to talk with patients has added a whole new level of motivation for working on these kind of problems. All right, first talk of the morning. You guys all know more about myasthenia gravis than I do, but we'll start off with an introduction anyway. So MG, a disease of compromised neuromuscular transmission. It is a prototypical antibody-mediated autoimmune disease where B cells generate autoantibodies that target skeletal muscle proteins. These antibodies cause a wide spectrum of muscle weakness, often beginning with ocular muscle weakness and potentially progressing to life-threatening respiratory failure. Something like 85% of these pathogenic antibodies that cause myasthenia gravis target the muscle acetylcholine receptor, and that's going to be the focus of the talk today. And then here, just some images of the kind of progression of disease. Okay. So neuromuscular junction, some basic anatomy in cartoon format. We have a motor neuron and the muscle cell membrane at this famous neuromuscular junction synapse. When an action potential travels down the motor neuron, vesicles containing the neurotransmitter acetylcholine fuse and release. Acetylcholine reaches millimolar concentrations, binds to these postsynaptic receptors. These are ligand-gated ion channels. The ligand is acetylcholine, so acetylcholine binds. The channels open up. These are cation selective channels. Mainly sodium and some calcium go into the muscle cell. That depolarizes the membrane. It's the first step in triggering muscle contraction. So that's a cartoon. We're interested in sort of finer, more realistic details. So here's an electron micrograph that I thought was particularly pretty. So here we have two motor neurons. Here's one, and here's another, termini of motor neurons. And here we have striated muscle fibers down below. We can tell this is a muscle cell. So we have a motor neuron here, and it's full of these small, clear little circles. These are the synaptic vesicles that contain acetylcholine. These larger, denser objects, these are mitochondria, making the ATP in these termini it's needed to synthesize acetylcholine and package it into the vesicles. The postsynaptic membrane here on the muscle cell, you can see it has these beautiful folds, and this is typical of skeletal muscle membranes, and these folds allow for more dense, more surface area of the membrane, so you can pack in more ion channels there. You may be noticing these dark squiggly lines on the muscle cell membrane. These are alpha bungarotoxin molecules that have been radio-labeled, I-125 labeled, so they show up really well in electron micrographs. So bungarotoxin is a peptide toxin from a family of snakes that binds essentially irreversibly to nicotinic acetylcholine receptors, the muscle type of these receptors. So this toxin has been used for imaging purposes like this to instruct us where are these receptors at the neuromuscular junction. They're postsynaptic. It's been used as an affinity reagent to purify the receptors, all kinds of useful things. And it's used, for those of you who do diagnostics, in diagnosing MG. The radioimmunoassay looks at receptors bound to radio-labeled alpha bungarotoxin. Okay, so postsynaptic acetylcholine receptors. So what are these antibodies doing for people that have myasthenia gravis? How do they cause disease at this postsynaptic membrane? So there are three kind of cellular level mechanisms that we talk about. One of them is complement activation. And so here we have our receptor in the membrane. Here's an IgG. MG is driven mostly by IgGs. They're bivalent, so we have two Fab fragments attached to an FC fragment. Depending on where this binds on the receptor and its angle of binding, which we'll talk about a little bit later, it can catalyze interactions with C1q, the first step in complement activation, which can recruit the membrane attacks complex eventually and damage the muscle cell membrane. And from my talking to clinicians and patients, my understanding is complement activation is kind of what we worry about most in MG because it can permanently damage the muscle membrane, right? Okay. Receptor internalization. So I mentioned these IgGs are bivalent, so a single IgG can cross-link a couple of receptors, and depending on what the subunit specificity is, it can kind of daisy chain or cross-link many receptors, and this can bend the membrane, triggering endocytosis. The receptors go inside, they get degraded, and you lose your cell surface receptors. Finally, we have blocking of acetylcholine binding, and the proxy for this is often competition with bungarotoxin, which binds in the same place as acetylcholine, but it's much larger. So you can imagine that acetylcholine blocking the IgGs blocking acetylcholine binding would cause rapid inhibition of neuromuscular transmission, but it's been a bit unclear how common this mechanism is and how much it contributes to disease. All right, what about therapies? So, of course, we have complement inhibitors. We're going to hear a lot about the different approaches to treating MG in the rest of the talks today. Other approaches, we have cholinesterase inhibitors like mestinon or pyridostigmine that broadly increase the amount of acetylcholine. Steroids suppress inflammation. Neonatal FC receptor antagonists can decrease the amount of circulating IgGs. Things like rituximab can kill off B cells. And in MG, thymectomy is commonly used that can reduce autoreactive B cells. So these therapeutic approaches work for a lot of patients. It sounds like it's idiosyncratic. You need to figure out what works for a given patient. It's very personalized, and it can change over time as the disease adapts or changes. But what I'd like to kind of point out here and what drives my interest in taking our structural approaches to understand this problem is that all of these therapeutic approaches can be thought of as broadly immunosuppressive, I guess, except for maybe increasing acetylcholine with cholinesterase inhibitors. But these would be expected to have off-target effects, of course, and the dream would be to have something that's more targeted to the pathogenic autoimmune antibodies interacting with a given patient's receptor. Okay, so how are we going to get to that? So a little more detail on the receptor itself and some biophysics. So here's the receptor in the membrane. These are ligand-gated ion channels, as I mentioned. They go through an activation cycle or a gating cycle. So in the absence of acetylcholine, these receptors are not conducting. Their pores closed. They're in a resting state. Ions are not going through in a resting state. Acetylcholine comes into the synapse. The receptor sites get saturated. The channel opens, so it transitions from a resting to an open channel state, conducting ions. If acetylcholine stays around for a while, tens of milliseconds, the channel will snap shut in the presence of agonist, and that's a desensitized state, so agonist-bound closed state. And this is thought to be a protective mechanism to not have too much calcium coming into the muscle cell. When acetylcholine concentrations drop, usually because of hydrolysis by the enzyme acetylcholinesterase, acetylcholine comes off, and then the channel closes, reverts to a resting state, and you get rapid resetting of this synapse, so you can get reproducible muscle contraction. So that's in a healthy neuromuscular junction. What happens in MG is you have these pathogenic antibodies that come in, and they can do all kinds of things that I mentioned in the last slide. So we have blocking antibodies that could directly compete for acetylcholine binding. We could have antibodies binding that activate complement, and we can have antibodies binding that cross-link receptors to cause internalization. All of these mechanisms can contribute to channel dysfunction. We know from recent work from Kevin O'Connor's lab that a single one of these antibodies can cause one or multiple of these mechanisms, which can give rise to some of the heterogeneity that we see in symptoms in patients, perhaps. Okay, so what are the outstanding questions that we would like to address? If we could get structures of this neurotransmitter receptor with antibodies bound, what could we say? So first of all, we would be immediately able to map receptor epitopes, which for the most part has not been done. It's very labor intensive, but this information would fall out directly from looking at a three-dimensional protein structure. What would we learn from the receptor epitopes? If we map them from a number of patients, we could start to discern whether there are any conserved hotspots for IgG binding on the receptor. You may have heard of the main immunogenic region. If that were to be borne out, then you could think about blocking or covering that, protecting that specific epitope on the receptor to treat disease in a very targeted way. We're also curious how common direct interference is with receptor activity. Most people don't do electrophysiology on these receptors in the presence of these antibodies, and we'd like to figure that out. And then lastly, can we start to understand how a single antibody can trigger a specific mechanism, say activating complement or causing internalization, or how can a single antibody cause multiple mechanisms? Okay, so those are the goals, and I'm going to address all four of those points today. The group that worked on this is Wan Li, a postdoc in my lab who did all the biochemistry and structural biology, Jinfeng Tang, who did the electrophysiology I'll tell you about, and Colleen Noviello, who is a project leader in the autoimmune disease area in my group. And then we partnered with Kevin O'Connor's lab at Yale and Min Pham, who was a graduate student and is now a PhD, and they had characterized a panel of monoclonal antibodies from MG patients in cell-based assays and then shared those with us and taught us some immunology along the way. Okay, so our approach. So the first major problem here is, why don't we already have information on receptor antibody complexes? It's because no one had been able to make enough of this receptor protein to get high resolution structures of it. So that was the first major barrier to try to get through. So I'll tell you about how we did this. We developed a cell-based purification method for the acetylcholine receptor. We then use cryo-electron microscopy to determine structures. We used to use crystallography. That used to be the go-to method for obtaining high-resolution structures of proteins, but that has changed in the last 10 years, and I'll tell you how it works briefly. Once we have reference structures of the receptors, we can add antibodies and determine their structures bound to the receptor. We then analyze these structures to see where do they bind and do the antibodies cause changes in the conformation of the receptor. We will generate hypotheses from the structures about how the antibodies might be pathogenic, and we can test those hypotheses by electrophysiology, and finally tie it all together to connect what we see to mechanisms that might underlie disease. Okay, so the challenge of making these receptors in a lab, even though this muscle acetylcholine receptor is the protein that gave rise to the concept of cell surface receptors in the early 1900s. We as humans have been studying this receptor for well over a century, and yet there were no structures of this muscle acetylcholine receptor. So why is that? So the correct assembly of this receptor, first of all, it's a pentameric ligand-gated ion channel. It has 5 subunits of 4 types. So there's 2 alpha subunits, an epsilon subunit between them, a beta subunit, and a delta subunit. So it's complicated. This arrangement that you see on the bottom is the only correct assembly, and it's the one that's found at the neuromuscular junction. It has 2 binding sites for acetylcholine at alpha epsilon and alpha delta subunit interfaces. Great, So when you try to produce this receptor, overexpress it in mammalian cell lines, what you get is every possible combination of these, and it's a huge mess. And most of them are not functional, not physiologically relevant. So we had worked on this for a decade, failed. Many labs had worked on this. It was an unsolved problem. But Wan, this postdoc, said he wanted to give it a try. He was very gifted, and he went through and he figured this out. So what he did was he made expression constructs, including the 4 subunits, in every possible arrangement. So he made a construct with all 4 subunits in a row. He made individual ones that he transfected together and separately. What ended up working was a pair of DNA plasmids. One of them is here, so circular DNA, and another plasmid is here. And this one has an alpha subunit gene and a delta subunit gene separated by this T2A. This is a self-cleaving peptide sequence. So when the protein's synthesized, it cuts itself between alpha and delta, liberating the 2 subunits. And then beta and epsilon similarly arranged in the second plasmid. So this ended up working. We made stable cell lines of these. After we got this to work, someone in the lab asked the question, I wonder how these are arranged in the human chromosomes. And so we went and looked at this, and indeed it actually matches. The only combination that worked for us matches what's present in the human chromosome. So alpha and delta are paired on chromosome 2, and beta and epsilon are paired on chromosome 17. So it sure would have been nice if we'd thought of this beforehand, but we did not. Yeah. So some biochemistry. Most of you are not biochemists, but I can't help myself. So this is a size exclusion chromatography trace. So we have protein amount on the y-axis, elution time off this column on the x-axis, which corresponds to the size of the protein. What we want to see here is a single sharp peak, which says we have a homogeneous protein species that's about the right size, and that's what this is. It's the human adult muscle receptor. And then this is SDS-PAGE, a protein gel showing 4 bands. We confirm by mass spectrometry that these are the 4 subunits in the receptor. Okay, great. Okay, so how do we take this purified protein and get structures of it? I mentioned we don't do crystallography anymore. Crystallography of membrane proteins is very challenging because membrane proteins have hydrophobic parts that sit in the membrane. You have to somehow keep those soluble, usually with detergent. Detergent interferes with crystallization. So about a decade ago, there was a revolution in cryo-electron microscopy hardware and software that allowed all of a sudden to get near atomic resolution structures through imaging using an electron microscope. So we have in our column of our electron microscope, we have an electron beam. Electrons are hitting our samples. So this is our protein molecules in ice, and the electrons come through and we get a 2-dimensional projection image of these 3-dimensional objects. You may see these cylinder-shaped things are oriented randomly, and that's important. You need 2-dimensional projection images that represent all of the orientations so that you can then reconstruct a 3-dimensional shape, which is the structure. Okay, so we then box out these individual images. This is done somewhat automatically, and we can then align these 2-dimensional images, the ones that are similar to each other, and when you average noise, like in the background here, noise is random, so it goes to 0, and the specific signal is amplified. This allows you to get a sense of data quality and also get rid of garbage that you automatically picked accidentally. Once we have this curated set of these molecular images in 2 dimensions, we align them to each other in 3 dimensions with increasingly finer sampling angles. And the more precise you can align these 2-dimensional images in 3 dimensions will determine your resolution of your structure. So if your sample's homogeneous, your imaging conditions are fantastic, you can get to the point where you can see individual atoms. It's incredible. Okay, so I'll now show you what that looks like in terms of the receptor structure. I told you this before, the receptor goes through this activation or gating cycle. What does that look like in terms of an electrophysiology experiment? So this is patch clamp recording where we have time on the x-axis and current on the y-axis. So we have no current here. We apply acetylcholine. Channels open really quickly. And then when we keep acetylcholine around, they start to desensitize, and we wash it off. The current goes to 0. So we wanted to determine, before we add antibodies, we wanted to determine reference structures of this receptor so we could see what the antibodies do to the receptor. So we got a resting state of the receptor and a desensitized state of the receptor in the presence of acetylcholine. So what does this receptor look like? Here is this extracellular part. This sticks out into the neuromuscular junction, into the synapse. So acetylcholine binds here, and it triggers a conformational change that opens up a hole through the membrane. This is a side view. Here's a top view. So this is the arrangement we were hoping to get. 2 alpha subunits with an epsilon between them, a delta and a beta. This little schematic here, this is the skinny part of the ion permeation pathway through the membrane. So that permeation pathway is defined by alpha helices, 1 from each of the 5 subunits. I'm showing just 2 here for simplicity. And these little dots, this winding pathway through the middle, those are not water molecules or ions or anything. It's just to give your eyes a sense of the shape of what the pathway would be for ions to go through. And red here means, dark red means it's too tight for anything to go through. So this is a closed resting state of the receptor, and it's pinched shut by hydrophobic amino acids. When we add acetylcholine, the channel opens briefly and then desensitizes. We're trying to study the structure of an activated state just for our own curiosity, but it's not stable at equilibrium. So what we get is a desensitized state when we add acetylcholine. That allows us to map the acetylcholine binding sites. This semi-transparent kind of background around the molecule, that's the experimental, what we call density map from cryo-EM. We're absolutely confident in how to position these molecules in this density. Okay. The ion pore conformation is a bit different. It's wider, but it's still closed. This is acetylcholine is bound, but the channel is non-conducting. It's desensitized. Okay, so now we have the foundation. We figured out how to get the receptor structure, and we have reference structures in the 2 non-conducting states that these antibodies might stabilize. So what's next? Okay, so we want to get structures with antibodies. IgGs are bivalent. They have 2 Fab fragments. If we add these to our receptors, we will expect them to cross-link and then precipitate, and we will not be able to get structures with them. So rather than expressing the whole IgGs, we make just the Fab fragments. Which Fab fragments? So we worked with Kevin's group here. They had, as I mentioned, Kevin O'Connor's group at Yale, they had profiled a bunch of monoclonal antibodies from patients and put these through cell-based assays to figure out, okay, what do they do? Do they activate complement? Do they cause internalization? Do they block bungarotoxin binding? And so we selected 6 of these that sampled different mechanisms. Some only block bungarotoxin binding, some do all 3 mechanisms, et cetera. So sampling diversity. So Juan put these into his structural biology assays and got 6 different structures, all at super high resolution. So the first row here is just the arbitrary naming of these Fab fragments from the IgGs. So 1B, 2, 3, et cetera. The next number is the resolution. These are all super high resolution. Don't worry about it. We have the top view of these receptor antibody complexes and then the side view. So this is the extracellular domain you saw before, transmembrane domain, intracellular domain. So as you glance around this very colorful, complicated slide, what you may notice is what we noticed first is that these antibodies bind all over the place. There's a tremendous amount of diversity from patient to patient. So each one of these comes from a different patient. Diversity in number of antibodies per receptor. So this one here, Fab1b, there's 2 Fab fragments per receptor. Same with Fab2, but the rest of them, it's all just 1-to-1. So there's 2 alpha subunits. It makes sense then that Fab1b and Fab2 bind primarily to alpha subunits. Fab1b causes all 3 mechanisms. Fab3 really only blocks. It's the best blocker of all of them. Fab6 and Fab7 don't block. They cause cross-linking. They don't activate complement. So we could spend hours. I could spend hours telling you about all these details, but I'm just going to pick a couple of classes of these to give you examples of what we learned. So we'll start with Fab3, and this one blocked, but it didn't do much else. And it's the best blocker of bungarotoxin binding of all of the Fabs we looked at. So where does it bind? So here's the receptor. Now we're looking at the atomic model we built into the cryo-EM map. So here's the Fab, and we're going to look at the specific interaction. So this semi-transparent surface is the experimental cryo-EM map, and then I'm showing you the atomic model that we built into it, which we often call the protein structure. One thing I'd like to point out here is the resolution allows you to position these amino acids in this interaction region with super confidence. Okay, so we have this alpha subunit and a delta subunit, and then we have the heavy chain, the variable domain of this antibody. This is one of the 2 acetylcholine binding pockets. It binds at alpha delta subunit interfaces. Most of this antibody interacts with the delta subunit up here. This is the delta subunit. The vast majority of the antibody interacts with the delta subunit, but there's a large insertion in CDR3 of the heavy chain, 29 amino acids, that works its way all the way down into this neurotransmitter binding pocket, where it would sterically directly compete with acetylcholine binding as well as bungarotoxin binding. So we saw this and we thought, okay, it looks like this antibody would probably then interfere with ion channel activity. We'd like to test that hypothesis. So we are making the protein for structural biology in a mammalian cell line. We can use that cell line for patch clamp electrophysiology and expose the cells to acetylcholine, measure currents. We can then add antibodies and see what that does. So here's a patch clamp electrophysiology recording. We add acetylcholine. We see this activation. We co-apply Fab3, this Fab fragment with acetylcholine. Nothing happens. And we thought, okay, well, maybe the antibody's binding more slowly. Acetylcholine's small. The antibodies are big. They have slower on rates. So then we tried pre-applying the antibody for, let's say, 15 seconds before we add acetylcholine. And then we see this big blocking effect. We wash off both of these, and we come back and we hit it with acetylcholine again, and the current stays small. So we conclude from this that these antibodies, indeed, they're slow to bind, they do block effectively, and they're slow to wash off. And if you imagine the context of a patient neuromuscular junction, these antibodies are around for a long time. So they are creating a steady state block of channel function, directly interfering with the channel activity, in addition to complement activation, internalization, et cetera. So acetylcholine 2 micromolar, that's a low concentration. What about a higher concentration like you might see during synaptic transmission? So we find, indeed, at both concentrations of acetylcholine, we get block of these currents. Okay, so that's Fab3 example, the best blocker. Let's look at this second class I'll tell you about, which is represented by Fab6 and Fab7. Okay, so Fab6 and Fab7 don't stick off to the side like we see most of our antibodies binding. Instead, they sit right on the top of the receptor. We've looked at a lot of antibodies, many of which we've generated ourselves as for tools, for GABA A receptors, for acetylcholine receptors. We've never seen anything sitting on the top of the receptor like this. It's really funny. This Fab actually makes contacts with 4 different subunits in the receptor. It doesn't touch the epsilon subunit, but its epitope involves 4 different subunits. So it sits on the top, and it has an insertion that reaches down into this, what we call the extracellular vestibule. It's kind of the beginning of the ion permeation pathway. Fab7, very similar. Sits on the top, and it has this insertion that reaches down into this vestibule. Very strange. So in Fab6, this long insertion arises from N nucleotides inserted at the VD junction, and in Fab7, it's in CDR3 at the DJ junction. So both of these long insertions have an arginine residue, which is positively charged, and a tyrosine residue at the tip. Okay. So some conservation here. So these 2 antibodies, they arise from different gene ancestors, they come from different patients, they have different subunit specificities, and yet they seem to share this conserved mechanism. We've never seen this kind of thing before. So let's look a little bit more at how this antibody might be interfering with channel activity. So here we're looking at the cryo-EM map. Hard to tell what's going on here. We have this alpha and epsilon subunit. We have the antibody over here sitting on top. And it's very dark in here, and I'm showing it that way kind of on purpose. So the ions need to go into this hole in the top of this extracellular vestibule to make their way down to the pore to conduct and depolarize the muscle membrane and trigger contraction. It looked to us like this antibody sitting on top, both for Fab6 and Fab7, might be plugging access of ions to this permeation pathway. So to look at this in a little more detail, here we're coloring the surface of the receptor by electrostatic potential. We're slicing through it so you can see the pathway in this vestibule. It's colored red because it's electronegative. It's a cation channel. The negative surface concentrates cations to enhance permeation. I told you before on the last slide that we have a positively charged residue at the tip of both of these insertions in the different parts of the antibodies that reach down, and they make electrostatic interactions, charge-charge interactions with this negatively charged vestibule. So we thought, this is interesting. These appear to both plug the vestibule and neutralize some of the electrostatics in the vestibule. Maybe they do something mechanistically different, but the result might be the same as with Fab3. So we wanted to do some more electrophysiology. If you if you make it harder for ions to get through, you could affect something like single channel conductance, how many ions go through per second, right? You make your hose diameter narrower, it's harder for things to get through. So here we switch to a different kind of electrophysiology experiment, single channel recording, where we have a glass pipette and we press it up against the cell membrane, and we hope to trap 1 or 2 or 3 at most channels in there, and we can then sample very frequently and watch channels open and close in real time. It's super cool. So here's what one of these recordings looks like. So we have this baseline, C is closed, O1 is one channel is open, O2 is two channels are open at the same time. So these numbers on the left are the holding potential that we're holding the membrane at, and we're looking at current. So we have a step here, that's one channel opening, a second channel opening, closing, both of them closing, etc. We measure these at different holding potentials. You can think of this as the electrical driving force, which allows us to calculate the conductance using Ohm's law, V equals IR. This is in the presence of Fab. So absence of Fab, presence of Fab. Fab 6 and Fab 7 do kind of the same thing. What you might be able to see, looking at these qualitatively, is that the amplitudes for these single channel events for a given voltage are smaller when we add the antibody. So the amplitude here is smaller than over here, amplitude here is smaller than over here. We can quantify this, normalize the current to the voltage to quantify conductance. In the absence of Fab, it's something like 42 picosiemens, and it decreases by about 25% in the presence of Fab. So these antibodies bind on the top. They plug part of the permeation pathway, make it harder for ions to get in. They also offset some of the helpful electrostatics in the permeation pathway, decreasing the favorability of concentrating cations, altogether working to decrease conductance through these receptors. So that's Fab 6 and Fab 7. I told you how Fab 3 also inhibits channel activity directly. We found that for all of these antibodies we studied, the Fab fragments directly inhibited channel activity to varying degrees. And so I think this was an underappreciated component of how these antibodies could contribute to disease, this direct interference with channel function. Okay, so how are we doing? Great time. Okay, so that's enough on the individual receptor structures, enough detail for this morning. I want to talk about the epitope landscape next. So what we're looking at here is a superposition. I've just overlaid all of our receptor structures with the Fabs bound, one copy of the receptor, but all the antibodies to give you a sense of where do they bind, how much might they overlap, are there hot spots or not. This is a top view. It looks like a big mess. It's hard to know what's going on here because there's no symmetry for your eyes to pick out. If we turn this on its side, you get this blooming bouquet or ice cream or something like that, whatever you're feeling, you can start to see from, and then this is just a different rotation angle to show you the different antibodies and where they bind. Some of these would compete with each other, like Fab 1b and Fab 2, they bind to the alpha subunits, they would compete with each other, even though their epitopes are distinct. And then some of them do not compete with each other. So we can then start to quantify popularity of subunits. Are there hotspots in the receptor? So here we're looking at I've broken out individual extracellular surfaces of each of the 5 subunits in the pentameric receptor. So here's one of the alpha subunits, here's the other alpha subunit, epsilon, delta, beta. The darker the color in red, the more commonly we see those amino acids interacting with the pathogenic antibodies. We can then make a table of these and see that alpha subunits are contacted by many of the antibodies. There are 2 alpha subunits, so there are more epitope contacts in alpha subunits, but delta and beta are commonly attacked by these pathogenic antibodies. Epsilon, much more rarely. We rarely see contacts with epsilon. If people are curious, we could talk about that. Okay. So the alpha subunit holds more epitopes. Most are in the beta sheet region of the extracellular domain. The MIR is not attacked. Okay, so what's the MIR? This main immunogenic region. Some of you will have heard about this, some of you maybe not. But early studies on MG by Socrates Sartos and John Lindstrom did really elegant work using an experimental myasthenia gravis antibody that's called MAB35. They generated this antibody in rodents by injecting the rodents with muscle receptor-like protein from the electric organ of the torpedo ray, very useful reagent. And it created symptoms in the rodents that looked like myasthenia gravis. And then they isolated antibodies, cloned them, came up with this MAB35 that was their experimental kind of tool to understand myasthenia gravis. And they found that many or most patient antibodies would compete with binding of this MAB35. And the conclusion from that was that that most of the antibodies bound in a common region. They then did very elegant epitope mapping experiments to figure out where does this MAB35 antibody bind, and it binds right here. So there's 2 alpha subunits that are identical. So this is the MIR on this alpha, here it is on this alpha. We see 0 contacts here. Now we've only looked at 6 antibodies, but none of them contacted this so-called main immunogenic region. So we would say maybe this is not the main immunogenic region. Maybe there is not a main immunogenic region, but their experiments were very careful. So how can we reconcile these apparent conflicting inferences? So the answer is these antibodies are big. The IgGs are very large. They're bigger than or similar size to the receptor's extracellular domain. So an antibody that binds here and an antibody that binds nearby will compete with each other, just not for the exact same epitopes. So I think we can all make sense of that. It's pretty clear and simple to understand. Why does this matter in terms of therapeutics? Well, if you're thinking about shielding a region or using some three-dimensional epitope to try to raise, CAAR T cells against or something like that, if you use the MIR, it wouldn't be very effective, at least in these 6 patients that we were studying. So I think it's relevant. The last point here is that many of these antibodies bind across multiple subunits. So again, from a therapeutic perspective, if you want to develop tolerance or make a CAAR T reagent, if you use just a single subunit, it's probably not going to be very effective. Ideally, you would make the whole receptor extracellular domain and develop tolerance to that. That's something we're trying to do. Okay. So how can we use this as the last thoughtful slide here? How can the structures predict broader pathogenic mechanisms? Something we think about a lot is complement activation, and we have mostly speculative ideas here. But we have antibodies like Fab1b. This one very effectively, the IgG form of it, very effectively activates complement, creating potentially permanent muscle membrane damage. So here's the receptor. Here's where Fab1b binds. From the structure, we know how the Fab binds. We don't know how the rest of the IgG would bind. But there are structures of IgGs in the databases, experimental structures that are very good. We can take those, superimpose one of the Fab domains from the IgG to generate a reasonably good confidence model of how the whole IgG might bind to the receptor. So the second Fab fragment for this one would point up, and the FC would be over here. And we just say here that this angle fits for complement. What am I talking about? So how do we activate this complement cascade? C1q assembles as this hexameric arrangement that needs to sit down on a hexameric platform of FC fragments. So these IgGs need to be bound at some kind of angle and some kind of spatial arrangement so that they can arrange in a hexamer that's favorable for this C1q hexameric assembly to sit down on it. And that works great in this cartoon. I'm trying to illustrate this here. So here's one IgG with its FC and these 6 receptors arranged around this kind of donut so that the C1q assembly can come and sit down on it, efficiently activate complement. So what about one of these antibodies that we tested that doesn't activate complement? So Fab 6 and 7, those are the ones that bind straight up and plug the vestibule, not at an angle. The ones that bind straight up, their FCs are going to point straight up, and you're not going to get this favorable arrangement. Now, there's qualifications here, of course. These are models. Also, I'm thinking mostly about IgG1s that have a small hinge region, not much flexibility between their FCs and their Fabs, FCs and Fabs. IgG3s have a longer hinge region, more flexibility, maybe more margin for error in activating complement depending on the binding angle. Okay, so these are things we're thinking about how to predict or understand how the antibodies cause different pathogenic mechanisms. Okay, to summarize, so these autoantibodies display heterogeneity in binding, everything. So they bind in different places, different spatial angles, different subunit specificities, different stoichiometries. All the antibodies we examined directly antagonize channel activity. So how much this contributes to disease, we don't know, but it seems to be a common effect. The structures can help predict the immunopathological mechanisms of an antibody. We can think about cross-linking. We can think about activating complement, certainly blocking acetylcholine binding. And then lastly, I'll say something about predicting protein structures. So there's been a revolution in this area as well. You may have heard of AlphaFold. It's gotten very good at taking away the need for us to determine experimental structures by computationally generating very good atomic models. It is terrible at predicting how antibodies bind to proteins. It's getting better at predicting protein-protein interactions, but the way something like AlphaFold works and these other AI mechanisms is they draw from databases on experimental structures. There were 0 experimental structures of muscle acetylcholine receptors with antibodies bound until 6 months ago, something like that, when we published these. So as we generate more of these experimental templates, that database will become more powerful, more useful in allowing us to predict protein structures. So we're very excited about this. Okay, so here's the lab. Here's Wan. Here's Colleen, who oversees the autoimmune disease areas, Jinfeng Tang electrophysiology, beautiful San Diego. Thanks to Kevin and Min, our collaborators at Yale, and funders, especially the MGFA, for getting us in touch with patients and bringing me here. Thank you very much.
We do have time for questions. If anybody has any questions? Maybe I can start up as people are waiting. This is unbelievable stuff. Don't think we hear about this very much in the myasthenia conferences. 2 questions. One is pure curiosity. All this stuff that you just showed us in 35 minutes.
Yeah.
How long did it actually take to get this done?
Yeah. It took Wan couple of years, which I would say is extremely fast. This guy has magic hands, good instincts. The labs have been working on this for 12 years. Lots of other labs have been working on it for many decades. So, we build off all this knowledge. It's been a big project. Yeah.
And the second question, this is complete naivety.
Sure.
Yeah, clinically, what we see in patients is, in some cases, we have patients who have different muscles that have weakness because of their myasthenia. As we treat them, some of these muscles respond very well, others may not. Could you hypothesize perhaps that there is changes that are occurring at the receptor level that might be responsible for this lack of improvement over time?
Let's see. Sure. So maybe we take the example of the droopy eyelids that I'm told we often see first. Those extraocular muscles have different kinds of neuromuscular junctions in them. There's some adult form of the muscle receptors, as well as those muscles are kind of special in that they also have some of the developmental isoform of the receptor, the fetal receptor. And the antibodies, because the subunit compositions are different for those 2 receptors. The adults have an epsilon subunit, the fetal isoform has a gamma subunit. So I could logically see there you'd have clear differences in how antibodies might bind to these different classes of receptors that could explain some of this. Yeah.
Hello, William Beck from California. Thank you for the wonderful talk. I just want to understand how your eye-opening presentation translates to the clinical setting. So as you know, clinically we order acetylcholine binding, blocking, modulating antibodies, as well as other types of antibodies, including MuSK. So based on what you're saying, I'm understanding that the blocking, acetylcholine binding, blocking, modulating antibodies bind to the alpha subunit. But if you look online, they're talking that they're binding to the MAR, but based on your presentation, it sounds like there might not be. And also, and regarding MuSK too, so we know that MuSK doesn't activate complement, and it's IgG4. So are you saying that the FAB is possibly 6 or 7? I'm trying to connect your presentation with the antibodies that we commonly order in clinical practice. So if you can comment on which antibodies bind to which subunit and which FAB class it would fit in, that would be great.
So before you step away, let me ask you for clarification.
Sure.
So when you say you order antibodies in the clinical practice,
Correct.
what do you mean you order them? Like you order some commercially available antibodies to use?
Correct, through blood tests, so LabCorp requests.
Okay, so you're placing an order to test for antibodies in patient blood, is that right?
Correct.
Got it. Okay, so great questions. So if the tests can look at competition with antibodies that are known to bind to the MIR. And you can see competition in that experiment even if they don't share an epitope, because they can bind somewhat nearby and then compete what we would say allosterically, where the FC domain or the other Fab domain would cause a clash and prevent binding even though they don't share an epitope. So those competition experiments are useful, but they can be a little misleading if you are concluding from them that, okay, these then bind to the alpha subunit and they bind in the same place. And we start to build up an assumption that antibodies with a certain activity all bind to the alpha subunit or the MIR of the alpha subunit. That may not be true. There are other ways these antibodies can, let's see, maybe I'll omit those details for now, but I think that's an important caveat in interpreting those results. As far as how can we relate subunit specificity to pathogenic mechanism, I'm not sure there's a great connection. I would imagine that antibodies that bind exclusively or mainly to alpha subunits, those are going to be more likely to bind to antibodies per receptor molecule. And in those cases, you can get profound cross-linking of receptors, because a receptor molecule has two antibodies that can bind to two additional receptors that already have antibodies that can bind to two more, and it'll cross-link kind of infinitely, whatever it can reach. Whereas if you have a receptor that has an antibody specific to an epsilon subunit, let's say, that can only cross-link two receptors maximally, because each receptor only has one copy of the epsilon subunit. So cross-linking internalization endocytosis, that pathogenic mechanism would relate to whether it binds alpha subunits or other subunits.
So of the 6 antibodies that you've run through the model, those were from 6 different patients?
6 different patients.
What is your conjecture? How many different antibodies might exist in an individual patient, and what happens over time? I guess you're planning to look at that?
Yeah. So from my reading, it's thought that MG is a polyclonal disease. And some of that, that inference comes from the heterogeneity seen in patients, that there's multiple activities. I don't think we know perfectly well. And so what we're trying to do to get at this in an unbiased way is use the receptor protein that we can make to fish out whatever the antibodies are against the receptors from patient serum and study this polyclonal mixture. And we've done this with a couple of cases where it's been successful, and we don't see a bouquet of heterogeneity. We see a single antibody bound from each of these patients. So I can only tell you about what we see, though, and not about what we might be missing. So I would expect there to be a range of affinities for these antibodies to the receptors. There are going to be some that bind very tightly. We will be able to preferentially detect those from patient serum. The weaker ones that might still be pathogenic are going to wash off in our experiments when we wash away non-specifically bound things, and we will miss them. So we're thinking about creative ways to stabilize receptor antibody complexes for the weaker binding ones, to try to give ourselves confidence that we're capturing everything, cross-linking receptors with antibodies before imaging them, that kind of thing. So we're very curious about this. We would love to map the heterogeneity within an individual patient. We're trying to do it. So far, it's working a little bit, but it seems like too speculative to conclude, but maybe there's an immunodominant antibody in some of these patients. Yeah.
Yeah. Thank you for this excellent presentation. I had a question about one of your slides. You show a patch clamp experiment in which there was a 25% reduction in the ionic current.
Yeah.
I was wondering, is there postulated the experimental level of significance of that reduction, and then trying to go from that to humans, knowing that their safety factor at the neuromuscular junction is 4 to 5-fold, that 25% reduction translate into impairment in that safety factor? Is there a postulation there of how to translate to humans?
Great questions, right. So healthy neuromuscular junction, these synapses are overbuilt. There's a big safety factor so that they always work, and there's tolerance for some imperfection. So I think this I'm guessing here, but I suspect this 25% reduction would not be enough to harm our ability, our healthy neuromuscular junctions to function well. But in the case of MG, where you also have some internalization of receptors, and maybe you also have some complement-mediated destruction, then this 25% reduction in current could become very meaningful. We'd like to test this hypothesis by using some kind of animal model where we make a lot of the monoclonal antibody and put these in where we've done a pretty good job of defining how we think they work, and then see what the extent of disease is that they cause. And if we used just the Fab fragment in, say, a mouse model of MG, then we would not be looking at complexities from complement activation and internalization and things. Just use the Fab fragment, which would allow us to selectively study the direct channel inhibition. So that's what we're going to try to do to test this kind of hypothesis.
And from a clinical standpoint, that would be an important question to answer in the sense that knowing a biomarker of what pathogenesis is active at the neuromuscular junction would help explain what therapy would be the most favorable, whether it's complement or whether ionic changes or otherwise. Thank you.
Thank you.
G'day, super sexy research. The single channel recordings, take you back to Fat and Cats and Quantal.
Yeah.
It's brilliant. So 2 questions. One is, do you think that there's any possibility that the antibodies, and I know you're studying FAB, so you can't tell just from that, whether the antibodies themselves could change the interchannel distance and therefore the packing density, because that's also a determinant of the summative end plate potential?
Sure. I'm sure they could. So these receptors are dynamic in the membrane, and they need to be packed at pretty high density, stabilized there by rapsin, et cetera. I absolutely would expect these antibodies to change channel density, mainly by bringing them closer together and stabilizing more tightly packing interactions. But that can be the initial step in also bending the membrane and triggering endocytosis. So,
yeah. And the second thing, you mentioned very briefly that the off kinetics of one of your antibodies was more than 15 minutes, and that's just the FAB. The whole molecule is going to have a slower dissociation content again. So that actually matters clinically, because when we do things like plasma exchange or FcRn, we're waiting presumably to get rid of antibodies going on, but we're also hoping to get rid of antibodies coming off.
That is a great point.
Yeah, so tell me more about that.
Well, I mean, that's...
Or is that a subject for another, another talk?
I'm just going to agree with you. I think that's really important to consider. Yeah.
Hi, we have one last question from our virtual viewer, which she's asking if you foresee any commercially available assays that could determine which mechanisms are active in a particular patient.
I don't know. We're certainly interested in partnering with people to try to develop assays. We do have one paper we published on a biochemical assay using HPLC separation to look at antibody binding that does not depend on competing with bungarotoxin or binding to bungarotoxin-bound receptors. That assay, the popular radioamino assay, can be a little bit problematic, because if your antibodies in your patient compete with bungarotoxin binding, then bungarotoxin will fall off, and you will not see that radioactive signal in the receptor, even though your antibodies are binding really tightly. So we do have an assay that doesn't use radio-labeled bungarotoxin, doesn't use radioactivity, but it does require an HPLC, which is, we're getting some pushback in the clinical labs. People don't want to have to run an HPLC. So we do have some ideas about diagnostics. And then in terms of, I love this idea of figuring out what pathogenic mechanisms does an individual patient have, and can we treat those rationally and get to what works for them more quickly. And then as the disease changes, also adapt to that. I don't know exactly how we're going to get there. It takes a while to go through all these kind of experiments, but I'd be excited to work on that. Yeah.
Thank you so much, Dr. Hibbs, for a fantastic talk, being on time and getting all these questions in. And it is 9 o'clock, so we're 1 minute early. So as we're transitioning, there's a whole bunch of seats interspersed everywhere. Can everybody who has a seat next to them raise their hand, please? For all those people out there, please come and help these guys out, because they can't put their hands down until you sit, and that's going to cause muscle fatigue. Fantastic.
Good morning again. Our next session is Biomarkers and Basic Science. And we will kick off this session with Dr. Punga.
Okay, good morning everyone, and I want to start by thanking the MGFA for actually supporting this work through a high impact pilot award. So this is the result from that grant now present that has been recently accepted. So we go now to biomarkers, which is something we really need for MG, and we need objective biomarkers to really predict treatment response and also subgroups in a better way. We also support monitoring of disease activity, and also we have a lot of different treatments coming up, but we need to know who to give the different treatments to. So this study aimed to validate previously found protein biomarkers as MG-specific biomarkers. So this was a multi-center cross-sectional study in Sweden and Germany, together with the team of Charité in Berlin. And we included serum samples from 200 AChR-positive patients, 192 matched controls from the healthy population, 93 patients with multiple sclerosis, MS, and 51 patients with chronic inflammatory demyelinating polyneuropathy, CIDP. And we wanted to do that to really see what of these protein biomarkers would be disease-specific and not just associated with neuroinflammation itself. So we used the system of Olink, which offers the proximity extension assay, and that, in difference to ELISA and other types of assay, offers to detect very small quantities of protein in serum samples. We did heavy statistical analysis with logistic regression analysis, PCAs, Baruta machine learning algorithms, etc., to really determine the strength of the potential biomarkers. And as I said, the work now has been recently accepted, so it's in press now in European Journal of Neurology. And what we found is that 14 of these proteins actually significantly distinguish MG from controls to start with. So this was a validation of the previously found data. And you see here we had the Bonferroni correction, since we did a lot of different testing of 92 different proteins, but these proteins still held up for being validated in MG versus the healthy controls. And with the highest one, as you see, Axin, ST1A1, IL-7. And then the very exciting thing was also the comparisons with MS and CIDP. And to the top, you see a heat map of odds ratios, and we corrected for sex and age in this study to really know what is the influence by sex and age. And there are some proteins definitely affected by that in neuroinflammation, as we know. But we can also see the different expression of the proteins in MG versus controls and these different diseases. And when you see the PCA down at the bottom and then translating that, so we did find a few proteins that were specific for MG versus healthy controls, and also comparing to MS and CIDP. And those were Axin1, ST1A1, STAMP-BP, CDCP1, and SIRT2. So we're very excited about these data to really distinguish MG from these other diseases. We also wanted to see whether we could find some proteins significantly defining subgroups in MG. And here we used again the logistic regression analysis. And in the A panel, we see a PCA of 15 proteins differing by MGFA clinical classification regarding the ocular, mild generalized, moderate generalized, and so on. So I haven't written out these proteins in detail, but they're in the publication that's coming out very soon. In B, we see the different proteins separating the different types of MG based on onset. So early onset, late onset, and very late onset. And we also saw very interesting increase in some of the proteins in the very late onset group, signifying that we have this increase in inflammatory proteins with age, inflammaging as it's called. In C, we see different proteins separating between the treatment-naive patients without immunosuppression and those that have immunosuppression. And in the D, sorry about the coloring here, but I think you can see it quite nicely. So in the D, we have these 10 proteins separating those that have standard immunosuppression, so more azathioprine, prednisone, etc., versus newer biologicals, which was FcRn and rituximab. So, in summary, I know 8 minutes is not a lot of time, so I thought to save some time for questions. So this study actually managed to validate these AChR antibody-positive MG, specific serum inflammatory protein biomarkers, AXIN1, STAMBP, ST1A1, CDCP1, and SIRT2, and we did find biomarker signatures correlating with the different subtypes of MG that I just described. This is severity, onset, and treatment response. So I would like to thank the team in Uppsala, Sweden, the Rubek Laboratory, and all international collaborators, and all the patients that have participated, which is very, very important for us to do this kind of trial, and the kind support of the MGFA and other funders. Thank you. Any questions? I didn't hear.
The timing of the samples.
Oh, the timing of the samples. So this was a cohort that could have been diagnosed many years previously. We also had a cohort which was recently diagnosed. So we put them all together, these 200 patients. So it's a big, big cohort. We're also looking closer at those that were recently diagnosed, but that's beyond this particular study. But the serum samples have been treated same and minus 80 degrees and so on, so they are quite stable in the freezer. Okay, thank you very much.
We have a slight change in our program. Unfortunately, Dr. Ji Lu couldn't be with us. So if Dr. Sanmarco is available and can provide her presentation, that would be great. Thank you.
Good morning, everyone, and thanks to the organizers for the opportunity to present our work. Today I'm going to show building of the mechanism of action of S1117, that is a seismic engineered pan-IgG protease. This is a disclosure slide. The enzymatic degradation of IgG offers a new therapeutic opportunity in autoantibody-mediated diseases such as Myasthenia gravis. At Seismic, we have identified and engineered S1117 from a highly specific, naturally occurring IgG enzyme called IDOS. We have used our IMPACT platform to remove but not eliminate B-cell and T-cell epitopes from this highly immunogenic protease to reduce immunogenicity. And we have also fused the protease domain to an IgG-Fc effectorless to extend the half-life. We have previously shown some data on S1117 to this audience, so today we're going to focus on different mechanisms that have not been shared. S1117 targets multiple pathogenic mechanisms in autoantibody-mediated diseases, so today we're going to focus on the cleavage of cell-bound IgG-Fc in tissue, how that reduced the complement deposition in tissue, and also the cleavage of BCR of the IgG B-cell receptor, preventing B-cell activation. To start off, And to analyze the IgG B-cell cleavage activation, we culture whole blood with S1117 overnight. The following day, we stain with flow cytometry antibodies to analyze IgG-Fc that is still expressed on the surface of the cells after cleavage, and anti-lambda or anti-kappa antibodies to see that when S1117 is present, those kappa or lambda IgG cells don't express that anymore, but they still express the IgG-Fc. So as we can see here, when S1117 is present, we see IgG-Fc, but lambda and kappa are eliminated in a dose-response manner. To go a little farther on what can mean that we're cleaving the IgG-BCR, on thinking of the disease indications that we're looking for, we did this study where we isolated untouched B cells. We cultured with S1117 overnight to allow for cleavage of the BCR, as I previously shown, and then we simulated antigen activation. So as we can see on the cartoon on the top, when the antigen is present, the IgG-BCR can bind the antigen cross-link and induce activation of these B cells that leads to phosphorylation of kinases, for example, SYK and ERK. On the other side, when the BCR is cleaved, so these cells don't have lambda or kappa, they cannot bind the antigen. Therefore, there is no intracellular activation, so no phosphorylation of these kinases. And we can see this on the histograms on the right. The intact BCR is depicted in red, and we can see the higher expression of kinases ERK and SYK phosphorylated, versus when the BCR is cleaved, so there's no expression of lambda or kappa, the cells have a profile activation similar to unactivated B cells. And we've done this in 4 individual donors, and we can see here how they behave. All the intact BCR in all these 4 donors have higher activation than when the BCR is cleaved. So what we're showing here is that the cleavage of the IgG-BCR by S1117 prevents the B-cell activation by antigen. Moving on to the in vivo part, we wanted to evaluate if S1117 can actually penetrate tissue and cleave IgG that is bound in the tissue. And to do that, we injected into B6 animals vehicle and irrelevant IgG control as an isotype control. Human anti-CHR IgG antibodies identified by Dr. Kevin O'Connor's lab. 1 week, and then the following week, we re-injected the same antibodies or vehicle control, but one group received S1117 IV. 2 weeks after, we collected tissue, in this case diaphragm, and performed immunostaining for IgG-Fc and mouse complement. And as we can see here on the stainings, the animals that received the human anti-CHR antibody have tissue-bound IgG. However, the animals that were dosed with S1117 don't have Fc fragments bound to the tissue and look very similar to the relevant control or even the vehicle control. We also analyzed C3 deposition, as I mentioned, and we can see that it localized very pretty in the animals that receive the anti-CHR IgG, so they co-localize on similar regions. But again, the animals that have S1117 that were dosed don't have IgG, so basically they have no C3 complement recruitment. So as a conclusion, we believe the IgG protease approach addresses multiple clinically validated pathogenic mechanisms and MG within a single molecule, offering advantages over existing therapies. We have previously shown that S1117 cleaves rapidly and sustained circulating total IgG, immune complex IgG, membrane-bound IgG, without affecting other immunoglobulin isotypes or other molecules that we have explored. And it reduces IgG-Fc-mediated effector function because it cleaves out the Fc portion of the antibody, therefore reducing ADCC and CDC. What we're showing today is that S1117 cleaves IgG-BCR, preventing B-cell activation by the antigen, and also cleaves IgG in tissue, reducing IgG-Fc by over 95%, preventing complement deposition in the diaphragm. And we're expecting S1117 to achieve dosing regimens that can be used treat-to-target approach that can be tailored to patient needs. S1117 phase 1 is ongoing. So thank you to all the people that have been part of this work, our collaborators, Kevin Nakar and Alexandra, and all of you for listening to me.